Cells, and what can go wrong. Warning, if you are bothered by a lack of drawing ability, then this video is not for you. As humans, we are made up of tiny things called cells. These cells are complicated structures that allow us to function as living things. Cells are made up of many parts called organelles, little organs. There's one organelle in the center of the cell called the nucleus. The nucleus is the control center of the cell. Inside it is DNA. DNA are strings of genetic information, basically the blueprints of a cell. DNA coils together with proteins to create things called chromosomes. Cells can also do this cool thing where they divide and multiply. There are a few reasons that cells divide: to help the person grow and to replace surrounding cells that have died. Cell division goes through 6 stages: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Together, all of those phases form the cell cycle. 4 of those phases specifically focus on nuclear division. Let's do a quick rundown of all the phases. Interphase is the longest phase, and in that phase, the cell grows and the DNA divides. Next comes prophase, the first phase that focuses strictly on the division of the nucleus, the process called mitosis. For this part, let's take a closer look into the nucleus. In prophase, the chromosomes condense together into rope-like structures and become visible for the first time. Next, in metaphase, the chromosomes move to the center of the cell in a row. In anaphase, the chromosomes split up and move to opposite ends of the cell. In telophase, the chromosomes on the separate sides of the cell form into 2 nuclei. Those are all the stages of nuclear division, but there is one last phase of cell division called cytokinesis. In cytokinesis, the cytoplasm of the cell splits. And with that, the cell has divided, and both new cells start over the cell cycle in interphase. The new cells can then grow and hang out as they are, or if needed, they can go through the cell cycle again if the body needs another cell to be created. But this cell cycle does not just conduct itself. It's orchestrated by many genes, notably the genes proto-onco and tumor suppressor. The proto-onco gene tells the cell when it needs to speed up the cell cycle, and the tumor suppressor gene tells it to slow down. These genes work together to efficiently control the cell. We've seen some amazing ways the cell works, but now let's look at what can go horribly, horribly wrong. Let's go back to that human full of cells. Let's say the sun, or a strange chemical, or any other mutagen, comes and starts messing with cells, or one cell. Let's say it affects just one cell. It scrambles up the DNA of the cell. This is called mutation. In a mutated cell, the proto-onco and tumor suppressor genes can break and lose control of the cell. When this happens, the cell spirals out of control and starts dividing rapidly, regardless of what the body needs. This is a crazed cancer cell. It divides and forms a mass of unneeded cells called a tumor. Once the tumor is big enough, it lets out a chemical that signals to nearby blood vessels. The blood vessels then grow to the tumor and provide it the nutrients it needs to keep growing. Blood vessel growth is called angiogenesis. Cancer cells from the tumor can break free and travel to other parts of the body, forming tumors in other places. This process is called metastasis. These tumors need to be stopped, or else they will keep growing and spreading until they begin to prevent other parts of the body from functioning. While we are still searching for a definite way to treat cancer, some possible but not completely effective ways to treat it include poisoning the bad cells, and also preventing blood vessels from reaching the tumor and therefore stopping it from growing.